Hey everyone, it's Jason, producer of Data and Biotech. Quick one, every episode of this podcast is now on YouTube with animated glossaries that break down the technical terms we discuss. Just search Data and Biotech on YouTube to watch. Welcome to Data and Biotech, a podcast from Cornell where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
James Yoder, welcome to the Data and Biotech podcast.
Yeah, thanks for having me, Ross.
Awesome. Well, just to kick us off, would you mind introducing us to your background and what led you down the path of drug discovery?
Sure thing. It's a little bit of a circuitous route. I studied statistics in undergrad at Harvard, and then I found myself working as a data scientist and started moonlighting, essentially as an ML consultant for a public biotech in North Texas, of all places. While I was doing some research trying to figure out how to build reasonable molecular property prediction models, a high school friend of mine who's a brilliant AI researcher put me onto using some message passing neural networks that Justin Gilmer and his colleagues at Google Brain published in 2017. And anyway, working with my now co-founder, Jim Thompson, we kind of went from there. The models worked well enough that two outsiders could sort of make an impact on small molecule drug discovery. And we're naive enough to start our own company on the basis of one anecdotal success. And then, OpenBench, the OpenBench dream began.
What I would love to do is understand a little bit of the history of OpenBench, because as I understand it, you've gone through a little bit of a business model change over the course of your history. So would you mind just talk about the founding story of OpenBench and what you learned and where you are now in terms of the model that you're using?
Yeah, so because I was a data scientist by background and my co-founder was a software engineer, we thought it was natural to start a software company, right? We were planning to do sort of molecular property prediction as a service, where we'd expose some predictive endpoints through a sort of ChemDraw interface, and medicinal chemists, computational chemists would go in there, draw their molecules, get predictions on any range of properties, and use those predictions to influence what they made and tested next. We ended up building basically a prototype of that software and selling it to a few biotech companies, sort of licensing it to them on a sort of pay as you use it basis. We pretty quickly realized it was not a very good business for us at the time. The models were so. It was very data hungry because it was a supervised learning system. So we needed a fair amount of data to train these models. And there was some literature data available, but not enough to make things interesting. Maybe we'll come back to this, but some of the federated learning approaches, for instance, that Toon Labs and Lilly are doing is very interesting to me because they have the data handy and they have sort of the consortium that they're building. So the models weren't great, and the business wasn't great either, right? It's a fairly shallow market. There wasn't a ton of willingness to pay at the time for biotechs buying sort of properties predicted on a screen. The understandable ethos was, why don't we just make compounds and test them, right? That's the way we've been doing things for a long time. It works pretty well. And, we found basically that the usage metrics just weren't that good, even for the companies that we signed on. So we assessed that, okay, this isn't going to be a viable business. We sort of went back to our initial pre-seed investors and we said, hey, what do you think? Should we give you your money back? We kind of hit a wall here. And they said, no, you're smart young men, figure something else out. And this was in the end of 2020, beginning of 2021, where it felt like money was free. And they were like, oh, whatever, you'll figure something else out. So we ended up pivoting into our business that we still run today. We hired a brilliant chief scientist, Lewis Martin, who's still with us, who had an actual science background in biophysics and computational chemistry. And he had developed some methods that we had basically seen his publication. He had put together a preprint on a way to sort of computationally effectively screen ultra-large virtual libraries and pick top hits without having to do sort of brute force characterization of every single molecule in that library. We liked the approach because it was very practical, very pragmatic to say, okay, we'll lose a little bit of precision here, but we'll be able to do this a lot cheaper. We picked up Lewis. We sort of figured out, okay, we have this interesting concept. How are we going to actually get people to buy it from us as opposed to all the other software players out there? Why don't we bundle this up into an operating model that could be attractive in its own right? And we sort of developed the success-driven molecular discovery model that is our sort of differentiated thesis to this day. This idea that, okay, don't trust that our technology works. We'll deploy it ourselves. We'll make compounds at our own cost and scientific risk. We'll test those compounds in your assays, and if and only if they work in those assays are you then on the hook to purchase the qualified hits from us. So that's a brief history of how we arrived, how we conducted this pivot in like the spring of 2021. And pretty quickly we found a couple of companies that were willing to take us on. So we sort of hit the ground running from there, and are proud to still offer the success-driven model today.
I want to unpack the success-driven model a little bit. So you mentioned that you're making compounds at your own cost and scientific risk. Can you walk us through what a relationship looks like with a new company that comes to you with a molecule or a set of molecules, and how you work with them to determine what the compounds are that they're then purchasing back from you after it meets some sort of standards or criteria for them?
Yeah, absolutely. So in general, what a company comes to us with is a target, and they've identified that target perhaps through their own platform, or perhaps they find it interesting because of other work that's been done against the target, or work that's been published in the literature. Folks come to us when they want that novel, potent chemical matter that can be the starting point for developing an asset that will eventually make it to the clinic. So oftentimes it's not a molecule or set of molecules they're bringing us, but rather just like the identity of a target that they think is promising from a therapeutic perspective, from a translational perspective. And what we do then is, our task is to find novel chemical matter that engages that target in an interesting manner. So, and to that end, we take a look at the target, and really we're looking at the structure of the target to see whether or not it's amenable to being drugged or being liganted is usually the first consideration. So can we find binders to the target? And to bind a target, usually you need some pocket to bind to, some groove, some cleft, some site that has concavity, that has basically opportunity to make interactions that would drive binding affinity. And the way that we assess these targets, there's nothing really special to it. We're looking at the same thing that any sort of medicinal chemist would look at, or drug hunter would look at, right? Is there opportunity to make barium hydrogen bonds? Is there opportunity for hydrophobic burial more broadly? Are there key interactions that'll drive affinity? So we conduct this feasibility diligence, and we try to be very pragmatic about it. Somebody sends us a target, they send us whatever structural information they have available. They send us maybe some literature that they think is the key literature around the target. In certain cases, they say, we don't actually have a structure of the target, but you guys can try to generate some predictive structures and work from those. That's increasingly common. Anyway, we do this assessment in 1 week, and we do it for free, so that, right, we can sort of abide by our success-driven promise, which is that the first dollar they spend is to purchase interesting start of chemistry, right? Something that's developable. So that is the first interaction, right? This feasibility diligence, once a target is disclosed under CDA. And usually, I'd say about 2/3 of the time, we come back to the prospective collaborator with a thumbs up, right? And maybe another 1 in 10 times, we sort of have a thumb sideways, where we say, okay, we'll need to generate some data to validate that this site actually exists. And then sometimes we just have to give a thumbs down because there's not sufficient opportunity to bind the target. It may just be that whatever, they have a protein-protein interface in mind that they want to drug that's very flat and featureless and not good to approach with a small molecule. So there's basically an assessment that we make, and we go from there.
I guess I'm really surprised at how frequently you're saying yes to people coming to you with proteins that are druggable. And like, I'm interested in, how do you handle the activity, and then also how do you account for sort of the frequency with which you're able to say yes with this success-driven model, given that a lot of drugs fail, is sort of the prior that I have there.
This assessment is just the first step. The next step usually is to consider, okay, we have a target. We think it's druggable or ligatable using our technology. What are the assays that are available to sort of confirm that experimentally, right? So there may be a suite of cell-free assays that we start with are, biophysical binding assay, looking directly at measuring binding of a small molecule to a protein target. Biochemical assay that maybe judges binding by saying, okay, we know if we bind to this site that we'll be competing with some endogenous substrate, and we can measure the competition to get an IC50 or a KI in an inhibitor, if it's an inhibitor design setting. So we next look at these assays. Okay, are the assays reliable oracles for specific engagement of the target? And if they are, then okay, second box is checked. We're that much closer to sort of striking a success-driven collaboration. The third major box is to design or define, I would say, the success criteria. So given the nature of the target, the binding site, the prior art, if there is any history on the target, we define success as a set of experimental and non-experimental criteria. The experimental considerations are usually potency first. Maybe we look at potency in a primary assay and some orthogonal assay. We want to make sure those corroborate each other. We want to potentially look for selectivity, or there's other aspects now of the target product profile that start filtering back to the hit profile that we're trying to achieve. There may be known off targets that drive toxicity. Okay, let's make sure we avoid those in whatever we're delivering. There may be certain things around mechanism of action, or maybe even a desire to confirm via structural biology. So let's get a crystal structure to confirm that this is indeed binding at the site of interest. So there's all of these, assays that we say, okay, these are going to work. And then we set thresholds essentially in each of these assays to define success. So oftentimes for a well-established target, we want sub-micromolar potency in some cell-free model. It's like a baseline threshold for success. If the target's never been drugged before, we might have a looser criteria for success. Okay, something that's even double-digit micromolar, as long as it's confirmed in multiple orthogonal systems, could be valuable as a starting point, acknowledging that there's ample medicinal chemistry that would have to be done to get that to a potency range where it could be a good drug. So there is a very case-by-case sort of bespoke success criteria development.
What I hear you talking about is that you're establishing a set of standards for what would be sort of a commercially viable or a thing worth purchasing from your partner in advance, and you're trying to establish the scientific criteria, the experiments that you would use in order to say, this meets those criteria, or this doesn't meet those criteria, because ultimately those criteria are going to be contractually governing in terms of whether your partner is obligated to purchase the compound back from you, and that there's also sort of if you lower the criteria that need to be met, then you could potentially lower the price that needs to be paid for the outcome. Because what I do think is data science relevant about what you're doing is that you have to assess upfront not just how likely is this to succeed, but what are the scenarios under which we will need to spend certain amounts of money in order to ensure the success of the compound that you're developing. Am I thinking about that right? Or, how would you update that?
Yeah, no, I think that's very accurate. And I think it is an interesting problem from a data scientific and broader scientific perspective, right? Like, how do we assess the risk of doing this? There's all sorts of what we call epistemic risk. That's not a phrase we've coined, but a phrase that exists to say, like, okay, we're embarking on a scientific effort. There's a lot of unknown here. If we fail, we're eating the cost of failure. We're not a big company that can afford to fail very often. We're trying to provide a good service. We can't, we have a reputation to protect. We can't afford to fail very often from that perspective. So how do we take all those risks? How do we pair those to our presumed out-of-pocket costs, right? Where the cost is also going to be associated with a risk, right? If we think it's a hard target, we're going to be making more compounds, we're going to be doing more characterization, right? So how do we mitigate those risks using technology? How do we improve our ability to predict these risks given our history running the success-driven model? And then how do we simplify all of those risks and costs into a single dollar amount that we can quote to a collaborator? That is essentially been one of the main projects of the success-driven model, is trying to figure out how to do all of that.
You were taking on all of this risk, but that also, I'm imagining from a potential customer's perspective, one of the downsides of entering this is that you're sort of you're exposing your IP to a potential partner without any financial transaction in advance. So I'm just interested, like, who are the types of companies of biotechs or researchers or members of the biotech ecosystem that are sort of ideal partners for you to work with, for whom the success-driven partnership model is a great opportunity for them as well as you.
The success-driven model drives us towards clients who are, broadly speaking, resource conscious, which these days is pretty much everybody in drug discovery, right? A lot of ink has been spilled over the fact that drug discovery is expensive, that it's slow, and people are looking for ways to use technology to try to improve the efficiency of drug discovery. And in our niche, we've taken a bite out of this by offering the success-driven model. The earliest clients were all in biotech, small molecule therapeutics, biotechs that may be pre-seed or seed stage, or they may be public biotechs. But in general, there's a lot of innovation happening where, okay, these are biotechs that are servicing new targets, that are willing to put money behind those targets, but also want to apply those resources in an efficient manner. And they say, okay, OpenBench, we have this new target. Nobody's drugged it before. You claim that you can target it for us, that you can find some start of chemistry. Have at it. And if you guys fail, we'll still have our budget to try a DEL or a high throughput screen or whatever, other more conventional method we could try. That's very common, right? Where we're sort of a first line screen. It's also common that people come to us after they've exhausted their conventional efforts, right? So they say, oh, we've already run a high throughput screen. We've already run, our internal libraries. We tried to do some work, maybe starting from literature starting points, but we don't have anything that motivating us to apply additional resources on hit to lead and optimization. So OpenBench, if you can meet the success criteria, we know that our bosses will basically give us the resources to keep working on this project. The board will approve of releasing the next tranche of funds to get this to lead stage. So that's another instance where we'll come in as like the back line. And that's always the most satisfying projects for us, where we can sort of pat ourselves on the back and say, okay, they weren't able to do this using conventional methods, but our approach was able to solve this problem. So for the longest time, and still today, our primary value prop that we're selling is, okay, don't pay us unless we succeed. Increasingly, we have a track record to say, work with us because we're able to do stuff that nobody else in the world is able to do, right? And we have this track record established through these previous projects. And that's pretty cool. And I think that will open up over time a different sort of market where it's less the cost conscious and more like the stereotypical pharma buyer doesn't care about saving $500,000, right, by accidentally running a screen that doesn't work. They care about finding that novel chemical matter that could someday become a blockbuster drug, right? So I think the track record will be a platform for us to sell to that sort of buyer in due time. We're trying to figure out a little bit on the fly how to appeal to that buyer, because we're so used to, whatever message discipline around the success-driven model. And some people just don't care about about that, right? About saving or reducing the money, or, running a screen that doesn't work. So anyway, I think the technology is broadly applicable, but the earliest market was the smaller biotechs.
Yeah, that's interesting. And it also strikes me that, because you're doing that initial screen for free, you're also giving information to potential customers, like just by engaging with you. So there's sort of an exchange of information. They're exposing the target to you, but you're also exposing to them, your evaluation of that target. Like, are you willing or not willing to undertake a success-driven collaboration that actually, produces the novel compound that binds to that target and meets all of their criteria. So that in and of itself is interesting. Before we move on from the success-driven model, would you just share sort of what your success rate is thus far? So, what percentage of collaborations have resulted in success? And then, if you have an assessment of what's your break-even success rate? So how successful do you need to be, like, over the long term in order to succeed as a company? I'd be interested in that as well.
The success rate that we've achieved to date is just a matter of fact. So we've now run 16 projects to completion with commercial partners, and 12 of those were successful. So 75%, to put a number on it. And those success cases, in the majority of those cases, we've sold more than one series. There's a fee for each series in general, is how we construct the agreements. So I think that is basically right in line with our target success rate across, thinking back to this large numbers, that's sort of what we aim for is scoping a project where the likelihood of success is around 80%. Though we try to be open-minded, right? Like if there's a chance of 50%, if we assess a priori that there's a 50% chance of success, we should be able to quote a deal to that success rate that's still profitable to us from an expected value, right, point of view. I wish sometimes that we just had like a standard pricing and we said, okay, we'll accept this amount of scientific risk for this set price because it would make our BD a little bit easier, right? If that makes sense, we'd be able to go to that first meeting and be like, this is exactly what it costs. But it's not a one-size-fits-all project because no drug discovery program is the same as the next, right? Over time, have been willing to take on more risk for higher prices, essentially. And there's new sources of risk that we find to be interesting from a market expansion perspective or from an impact perspective. It's always going to be more challenging to work from a predicted structure of a protein than an experimental structure. Predictive models have gotten a lot better. There's a new set of epistemic risks that those models carry. There's different sources of uncertainty of how a binding site is arranged that we're trying to model. It unlocks the ability to do structure-based screening against a wholly host of targets that haven't been crystallized before or that are defiant to being crystallized. And that's really cool, but it also means that we have more risk, right? So maybe over time, ironically, maybe our success rate will get worse, but our business will become more viable. I believe that could be true. And I think that in general, it's always painful to fail, but we have the ability to sort of sleep at night knowing, okay, we are bearing the cost of failure when we do fail, and we haven't charged an arm and a leg to our clients for that failure. Now, there's still time and effort that they put into these projects. Don't get me wrong. Like it's always painful to deliver or to sort of come to the conclusion that we failed on a project.
One thing about your model that's really interesting to me is that you seem to have come up with a model that sort of gets around the principal agent problem that you often have when you're doing partnerships with researchers or consultants. And I say this as a consultant myself. The thing that enables you to do it is that you've created this set of computational approaches to working with your customers that are sufficiently advanced that they give you the information you need to make decisions on the fly about which of these projects is worth working on, and also some information about like what is the path to getting there, because you need to also price out how much investment needs to be made in experimentation, in computational cycles, in people working on the project, et cetera. So to the extent that you can, I would just love to unpack, what is the end-to-end data engine that's setting you up to be able to do this kind of model. Let's start from the beginning, like from the very beginning. So you've gone through this feasibility study. You have, settled on the criteria. You've agreed on a price. You've signed a contract. Just from there, what is from a computational perspective, like what happens from end to end?
Yeah. So the first thing that we're doing is figuring out what are the models of the protein that we want to screen. So we're sort of setting up a horse race internally. Essentially, we're going to, at the end of a virtual screen, make somewhere like 600 to 1,000 compounds, right? We have a pretty good sense of what the library is that we're going to be screening. Typically, we're screening these ultra large virtual libraries, right? So there's libraries these days in the number in the trillions of compounds that are commercially available that have not yet been synthesized. So our preferred vendor for this is a company called Enamine, who are the global leaders in this space in terms of pioneering these ultra large virtual libraries for commercial use. And they are phenomenal partners. Shout out to our friends at Enamine. And they basically say, okay, here's a set of building blocks we have available and a set of reactions that we can run to combine those building blocks. And that's sort of half of the equation for the virtual screen, right, is that virtual library. So it's not even explicitly enumerated. To explicitly enumerate it would be like computationally challenging because it would be hundreds of terabytes of compounds in a flat file. And it's just not very ergonomic, right? It's not something that you can just sort of copy onto an E3 instance, right? Or really, a lot of what we're doing now is on the fly. So we're sampling basically reaction subspaces within this large chemical space, preparing to sample those in a way that we can sort of turn the crank on is part of our data stack. So ingesting these libraries from Enamine and similar companies and preparing them for screening is the first piece. I got a little bit ahead of myself, right? Behind that is figuring out the structures that we want to screen. So if we're working from a predicted protein model, there may be different snapshots essentially that we're taking, predicted snapshots that have, whatever, slightly different arrangements of the binding site. In principle, sometimes binding sites are rigid, but a lot of times they're sort of breathing, right? A protein is sitting in solution and it's moving all the time. These are very dynamic systems, but what we're doing to screen first at trillion compound scale is just taking static snapshots that we're going to model ligands into. So we're basically picking those snapshots. What do we think are most interesting? We actually run the virtual screen. So we basically say, okay, we're going to take these large virtual libraries. We're going to start sampling ligands from those libraries and modeling them in the binding site. In the binding site, we are basically generating a bunch of poses that this ligand could take and scoring those poses. And this is a concept that we did not pioneer, right? Basically, molecular docking has been around since the 1980s and has been worked on for a long time by people much more intelligent than us. And we haven't really reinvented the wheel here. There's some new methods that we use sometimes for pose generation. Increasingly co-folding methods can be useful. So like all atom protein ligand co-folding. But we also don't really do any research in that area ourselves. We're basically just receiving the state of the art, both in sort of more conventional docking and in co-folding to generate poses. And then we are using our own technology to score those poses. Or in certain cases, it's called rescoring. So essentially to try to use our machine learned methods to predict the affinity of a ligand to a protein if it occupies that pose. And the pose is important because the affinity is being driven by, right, all of these essentially physical interactions between hydrogen bond donors and acceptors, and the distance between a donor and acceptor matters a lot there's all sorts of interactions like that are driving affinity. There's certain, right, hydrophobic burial that's guided by how the ligand is actually sitting in a binding site. Is it displacing certain waters that may be in that binding site in solution? Things like this are driving affinity, and essentially that's what we're modeling. We're guessing could any given ligand occupy a certain pose, and what is the affinity that it would have in that pose. So we do these predictions at scale. We're sampling on the fly, sampling, canonicalizing, docking, rescoring. At a certain point, we basically feel like, okay, we've built enough of a sample of ligands mapped into affinity that we feel like we have a training set. We can use that training set to build then a proxy model. It's a lot easier to infer affinity using this model across trillion compound chemical space. It's a lot more computationally efficient to do that rather than to dock and score every molecule. We use this proxy model as a tool to infer and then search through the chemical space. So really what we're doing these days is we're looking through each reaction subspace to try to find the highest scoring ligands. And we use this proxy model to sort of prioritize ligands to feed into the scoring oracle. And what we see is, right, over a few and basically it's an active learning loop, right? We actually generate this sort of computational label, the score. We train a model using molecule into label, and then we deselect the next sample based on that inference, and we do it all over again, right? So we'll do that until we basically hit certain stopping criteria, where we'll see a sort of saturation of score after a few iterations. And then we have our highest scoring molecules across these different reaction subspaces. We end up selecting our favorite reaction subspaces and focus on those to start winnowing down, basically going from trillions to, right, hundreds that we're going to make and test. And all of this is basically happening out of curiosity, mostly using like Python scripts that are being executed from a shell script, right? Like written our own scoring functions in Python, essentially, that are trained, and we're using those trained weights then to, whatever, after we encode a protein ligand system, we're basically passing those encoding through a model to get that predictive affinity output. And then all of the active learning loop is also happening, basically using supervised approaches in Python. So it's pretty, we built this whole auto screen infrastructure is what we call it internally, so that we can make sure we're running stable releases, right? So we, reduce sort of any sort of technical risk. We have enough scientific risk, right? We try to mitigate as much as possible technical risk when we're undertaking these screens.
Would love to hear from you. What have you all learned about searching these massive libraries and finding the subset of compounds that are going to be best both for like computational evaluation and then also for bringing together in order to train the scoring algorithm that you have further down the pipeline.
In general, when we design molecules, we're almost never making the molecules because we think they'll be good training data points, right? We're making them because we think they'll be good hits, right? We think there's a good chance that they are novel, potent, developable, and that they'll essentially satisfy the success criteria that we spoke about. It so happens that the vast majority of the molecules that we make and test in any given project, like I would say over 99% of the molecules we've made, are molecules that have never been made before and that generally occupy a chemical space that's not exemplified in the patent literature. Well, maybe saying the chemical space hasn't been exemplified is a little bit of an overstatement, but these precise molecules have not been made and tested before in the vast majority of cases. So there's sort of this nice byproduct, which is that, okay, if these molecules are active or inactive, we can have basically a label on those molecules, a label that was generated by predicting this property using our last iteration of the scoring function. We could take that and feed it back into the scoring function. So it's not just to address the way that you phrase that question, we're usually not doing search with the objective being like, okay, we're doing some sort of uncertainty quantification that's giving us that next molecule that's most effective. Sometimes in like the purely computational loop, that is happening, right? Where we're trying to surface molecules that we think are high, going to be high scoring via inference or that are high perplexity via inference. But when we're actually making compounds, it's usually because they're already high scoring. Now, a lot of those are inactive, right? The vast majority of the compounds we actually make are never been made before and they're inactive, right? So it's very useful to be able to feed those back into our training set, right? And to train new scoring functions that learn from, okay, it's like scolding a child who's done something naughty, right? You have to learn from example. You told us that these 100 molecules were really active and only 3 of them were actually active. Take a look at these 97. Take a good hard look at them and think about what you've done, right? By surfacing these. And I'm not a parent yet, so that's probably bad parenting advice to try to rub your child's mistakes in their face. But I think the analogy stands. We're always making molecules that are inactive, and to be able to integrate those into a training set is valuable. We've learned a lot about search partially by necessity, right? Over the course of OpenBench's existence, these libraries that we screen have gone from sort of billion compound scale, which was able to be enumerated in a matter on gigabyte scale, into trillion compound scale, right? And there's a form factor there that we've had to respond to, and that the industry more broadly has responded to. A lot of people are thinking about ways in which they can sort of, in reactant space as opposed to product space, the number of theoretically accessible small molecules that are sort of in a drug-like chemical space is beyond comprehension. It's in the order of like 10 to the 60 is what some people have said. And those people aren't just talking out of, they're not just, people have done these calculations, and, we're just capturing a very small piece of that's sort of low-hanging fruit from a synthetic accessibility perspective. But even within that low-hanging fruit, there's a possibility to fall into local minima if you don't stratify these spaces according to different reactions. In our experience, there's a lot of amide coupling reactions that occur and a lot of building blocks that are amenable to amide coupling in the enamine library. We're not the only ones that have observed this. There's tens of billions of amide couplings. Maybe you could end up oversampling those if you just take a random sample of chemical space and back and score it and then go from there. So doing this sort of stratified sampling and thinking about almost building an individual model for each reaction subspace has been a huge boon for us in terms of enriching hit rates and enriching the diversity of chemical matter that we're making and testing, which I think is a big part of the point, right? Like if you're screening trillions of compounds, you really want to be able to take advantage of the full diversity. And it may be that there's smaller, quote unquote, reaction subspaces that merely have tens of millions or hundreds of millions of compounds, but where there's really good chemistry that's really complementary to a binding site. It may be missed if you're not being judicious about how you're searching and sampling these massive spaces. Just because a simple random search of tens of millions out of trillions, even if you sample 1 million molecules, you might get only a couple of exemplars, a couple of representatives, and then you're training your proxy model on very sparse data for that particular reaction subspace. And you're not going to pick up on, well, at least our Oracle isn't good to pick up on that with such a small sample set. So hopefully that's making sense. It's a somewhat complex thing that we think about a lot. So I may be underspecifying the sort of shape of the problem and how we think about it. But there's been a lot of methods development and sort of searching. But yeah, a great thing about, and what makes our scoring function bespoke, and the great thing about the success-driven model more broadly, is that we are bearing this cost and scientific risk, and it basically creates a permission structure in which our collaborators, unlike the standard fee-for-service MSA relationship with a service provider that I spoke to earlier, our collaborators say, okay, you're bearing this risk, you're making these molecules at your own risk, you're paying to have them tested. We'll license back to you after we purchase these libraries. We'll license back to you the rights to use these libraries to improve your technology, as well as the data that's been generated in our assays. And that's really cool because now we've run, a couple dozen of these, and that's really high quality, purified samples tested in primary assays against a wide range of targets, target classes, binding sites. And there's certain global sort of biophysical principles that we're able to learn, right, with sufficient data, layering that on top of, right, high quality literature data that we've sort of pruned and cleaned up. In particular, it's exciting to know that there's a way to sort of, right, there's a data flywheel that's feeding back and trying to avoid those pitfalls that we've fallen into before.
It sort of leads to the potential hypothesis that as you do more and more of these collaborations, in addition to getting better at selecting the potential collaborations that are likely to result in success, you're also able to, because the scoring function of the libraries is sort of built on the data that you're creating as you're going through these collaborations, that you're also going to increase your own success rate by gathering that data. So I'm just interested in, are you already seeing that with the data that you've gathered so far? And what's your sense of how applicable one version of this for one client is to the next compound that you're developing for the next client?
It's a really interesting question. I think in principle, we can say with confidence that based on the benchmarks that we've developed, the data we're using or that we're training on from previous collaborations is improving our scoring function, right? And that improvement of the scoring function, in principle, exactly increases the chance that we succeed on the next target. The scoring function that we have is a global scoring function. So it's not something that is even fine-tuned on whatever literature data may exist for a given target. There's a lot of conjecture, really, around how our training data improves our scoring function from one project to the next. What we see in general is that, right, in 2021, we fashioned a benchmark using a basket of targets, right? Kinases, GPCRs, other enzymes, other receptors, whatever, a suite of targets for which there was ample literature in, here are molecules that bind these targets, right? So we basically took all those actives. People don't really publish their inactives that much. So we basically created a property matched set of decoys, inactives, and it's essentially a classification task, right? Like, okay, for a given target, is this molecule active or inactive, right? So we're able to generate classification metrics on this task, your classic receiver operating characteristic metrics. Okay, what's the area under the curve for a given scoring function, telling us whether it's able to differentiate actives from inactives. Then would look at is, okay, across these targets, for some of them we have high AUCs, 0.8, 0.9, 0.99 for certain targets because, right, our training data is enriched with structures, protein ligand systems that are close to whatever that target may be. So for example, if we're looking at a competitive kinase inhibitor or a type 1 inhibitor, right, there's a lot of crystal structures that exist in the literature that sort of show, okay, you need to make this hinge binding interaction to drive potency in most cases, and whatever. There's sufficient depth and diversity of training data to be good on those targets pretty much out of the box. But there's a bunch of other targets where we underperformed. What we've seen over time is that, yes, now today, basically our worst gauge performance across any of the targets is better than what our average performance was in 2021, in large part due to the data that we generated, cleaned up over the course of our collaborations, as well as improving our ingestion of data from the literature and improving the method itself, right? The actual algorithm and how we sort of featureize these systems or encode these systems. That's been a huge boon for us, I think, in terms of like the success, the enrichment of hits in the libraries that we make. I have good reason to believe that, right, that Oracle will continue to improve over time, right, our ability to predict affinity, which should be a positive feedback into, right, the way that we bear risk and our ability to, drive good value for our collaborators. So that's exciting, an exciting observation.
For sure. And the thing that strikes me about that is that if I understand correctly, the benchmark that you created prior to a lot of these collaborations. So it's not as if the benchmark is weighted toward the collaborations that you had. It's the benchmark is representative of all the potential collaborations that you that you might have had. And so what you're seeing is that the selective collaborations that you've had are improving your performance on a basket of collaborations that might have looked very different than the ones you actually had.
Yeah, exactly. And I think the way I'd restate that is like the targets we've worked on in collaboration do not overlap with the benchmark targets. I think actually just by coincidence, one target we've now worked on is in the benchmark, but that's one of many. And basically, yeah, it's a good sign that we're learning some we're learning something globally about these protein ligand systems that we're able to apply to the next target that we haven't seen before.
Yeah, awesome. Well, before we wrap, we've spent a lot of time on your model and on sort of the data science approach that you use to be able to enable that model. Would love to hear about the some examples of companies that you've worked with and sort of how those projects went.
We always have to be sensitive when talking about our collaborations, right? Because we want to protect the competitive interests of our collaborators. It's one small point of frustration for me because I think we do all this cool work, but we can't really show it to people because, right, it's not patented yet by our collaborators and they're trying to carry it forward, right? But there's a couple of collaborations I think I can speak to because we put out some press. One of those is with a company that was one of our first sort of, to whom we owe a lot, named Tavros Therapeutics. Tavros was a functional genomics platform that spun out of, I believe, some work at Duke, and they've since been acquired by a company called Vividion, which is actually they themselves are a Bayer subsidiary down in San Diego. So Tavros, though, their emphasis was really on the biology, right? They have this CRISPR platform surfacing synthetic lethal targets for application in cancer therapeutics, and they didn't have the chemistry expertise to launch these projects internally. They didn't have their own libraries, small molecules. They didn't have existing vendor relationships for high throughput screen or anything like that, nor were those screens something that they could prosecute internally. So we kind of found them at a good time where they were very cost conscious. They were very young, seed stage company when we first came across them in 2021. We had this sort of newfangled idea, and their founder, Owen, who I admire a lot and who's a very scrappy and effective entrepreneur, was kind of like, okay, we think we had good fortune where we had one case study we had developed internally, which was on the same target class that they were going after. So we said, look, we can do this. And we had actually some relationship with one of their consulting medicinal chemists prior, which was also good fortune for us. And they said, okay, we'll give this a shot. And they saw the appeal of the success-driven model. We gave them a pretty sweetheart deal because they were one of our first collaborators. We were still doing a little bit of price discovery. And it worked so well against that target that we decided to re-up and undertake a multi-target collaboration after the initial target. So I think it was, 2021, we launched our first target. We delivered some really nice quality chemical matter in 22, and then 23, we decided to take on multiple targets together coming out of their platform. So that was a great story for us because it showed that we were able to serve young companies that were very cost conscious and that we were able to be complementary to companies that were really strong on the biology platform where we have really no expertise. And despite not having expertise in their targets or their CRISPR tech, had our own operating model that could fit really well and be very complementary to them. And we know that they were able to take those molecules and improve upon them substantially, which is also encouraging, right? It's always um, a question that people have for us. We don't maintain any formal audit rights, but it's always encouraging to know, okay, the goal was to deliver something that was potent, novel, developable. We know experimentally that these are potent. We know they're novel just because we can reference the literature, but are they really going to be progressible? And that's a little bit of an unknown until you start chemistry. And they were able to make a lot of progress with the potency and selectivity of these molecules and getting, proof of concept in more advanced models than just the biochemical systems that we were originally testing them in. So that's, I think, another case on sort of the other end of maybe the spectrum, whereas Tavros was very small, was working with a company, Cure Therapeutics, with whom we were working with a really seasoned set of extremely good drug hunters, medicinal chemists and computational chemists and biochemists and structural biologists who had existing relationships with CROs and a lot of capabilities internally on the chemistry side. And they were just going after a really hard site that was essentially a novel site. And they saw OpenBench as an opportunity to sort of take a flyer and say, okay, you guys think you can do something against this difficult site? Prove it. And we did, which is awesome. And we were ultimately able to release that we found the most potent known chemical matter at that binding site of interest. Anyway, it was a nice feather in our cap when we were able to share that with the world. And these are scientists that we worked with for whom I have an immense amount of respect and who have been around the block. A lot of them came out of this sort of Array biopharma diaspora, and Array was doing a lot of this century has been one of the most productive sort of research houses. We're sort of a services company, then developed a pipeline, got by Pfizer and Boulder, and then in Boulder, a lot of their scientists went into the wind, and a lot of seeds sort of germinated all over Boulder, and we got to work with that crew who had done some really impressive stuff that we didn't even realize we had read their papers previously as cool work. So a different case, right? Working with a more established company, working on, where they had a lot of in-depth expertise around a target, but we were able to support novel discovery against a novel site against this target that they knew well. And it was a nice complement, a collaboration that I also cherish in my memory.
So as we head toward the end of our conversation, I'm just interested in hearing from you, how do you see the success-driven model evolving over the next 3 to 5 years?
Yeah, this is a $1 million question. Maybe someday $1 billion question. I don't know. There's a deep enough market for it to be $1 billion question, but there's within hit discovery, we've been able to make a lot of impact, but we acknowledge that we're just, taking this sort of first step on a 10-step progress. So there's a natural extension to deliver chemical matter that's even more advanced. We never set any in vivo criteria within our success parameters, for example, in an industry collaboration. So being able to bite off more and more, it's just in 2022, all of our success criteria were specified in cell-free assays. We started moving into cell-based assays, into early ADME. So sort of incrementally biting off a little bit more towards like what a target product profile might look like. Continuing to onboard those sorts of things, I think, is exciting for us. And maybe we'll end up being more of a lead discovery house than a hit discovery house. Continuing to get creative with our collaborators on ways to serve really, really young companies is an area of excitement for us. Right now, I think there's a lot of need for young companies to have like resource efficient discovery services available to them, especially because we're at like a low watermark in terms of seed and Series A funding and funding of research in particular. Trying to find a way to enable new platform companies, emboldening them to launch new projects, I think is exciting and something that we can do. There's a market that we feel like we can create there given the success-driven model and sort of solving the chicken egg problem where it's like, okay, we're not going to be able to raise money until we have chemical matter. We don't have to pay OpenBench until we have chemical matter. Maybe there's some where, okay, we'll be able to find that matter for them that they'll be able to raise on the back of. So exploring that almost down market of where we currently sit is also quite appealing. And then up market from us is like the lead discovery and working with more and larger and more established biotech and pharma companies as well is I think an opportunity. We see increasing opportunity to work with other CROs too. Like a lot of CROs have this incumbent fee-for-service business, but we can work with them directly. A lot of times the characterization that's being done in our collaborations is with CROs doing the doing the biochemistry, the biophysics, the structural biology work for confirmation. So we depend upon these CROs, and working more closely with them is an area of interest. We have a certain moral clarity around like sticking to our services mission, right? Really, we want to serve scientists, and there's a lot of AI companies that have gone before us. Like, we think we're good at hit discovery, and that we're so brash that we're willing to offer the success-driven model. We don't want to fall into the same pitfalls that a lot of companies before us have fallen into, where they're like, we have an edge on this small part of drug discovery, and so now we're going to develop a therapeutic pipeline. There's a lot of like, I don't know, reasons that venture-backed companies go in that direction, because there's an easy path towards like the billion-dollar outcome. If you have a safe and effective drug, right, and you're helping patients, then usually there's a billion-dollar outcome there. Services are a lot less sexy and a lot smaller market generally. So we're okay with that, though. I mean, that's the basis on which we founded the company, and I think there's a lot of opportunity for us continuing in the service vein. I don't know. I think trying to still figure out some of the details of the success-driven model along the way, right? Getting even better at risk assessment, getting even better at taking on more custom synthesis, right? We prosecute these ultra-large libraries. What's the next step there? Yeah, like improving our methods to justify synthesis where we're spending $1,000 per compound, right? Which is closer to the whatever order of magnitude you're spending in a more custom synthetic regime, is an area of great interest to us as well. So we're trying to do some methods development there with, branching outside of our existing ultra-large virtual screening. I think more broadly, we really believe in success-driven discovery as a meaningful theory of change for AI and drug discovery. Like, if AI and drug discovery is going to make an impact, what matters is the results, not the technology. And that's very much been our ethos from the beginning, inspired by others who have gone before us, who have sort of made that their ethos. There's a quote from Ant Nichols, who was a founder of OpenAI, who's talking about the importance of delivering, when in doubt, deliver results, not the tech. So I think continuing to plow that furrow will be ultimately fruitful for us.
Is there a single challenge or roadblock that you see to taking on a lot more collaborations, or taking on bigger collaborations and scaling up the work that you're doing as a services organization?
I don't think that these are insurmountable, but some of the roadblocks today, one is like, the more ambitious we get, the more capital we need. That's one risk. That's one thing about the at-risk model that we engage, is that we need working capital. So trying to figure out how to do that in a way that makes sense for ourselves and our long-term goal, right? Like, there is a little bit of a tension between, like, venture doesn't usually fund services companies. I don't know, how we capitalize the business is something that we're trying to figure out. We're revenue generating, unlike a lot of biotech companies as well. So, like, obviously we can bootstrap to a point, but if we will, and that's a lot of what we're doing, but at some point it may make sense to take on capital, and how we do that right is a big question. And I think we're outside of the box for a lot of, definitely for biotech VC, and even for sort of like tech bio, or like, tech VC. So trying to figure that out. And then, like, right now, I see that there's been a little bit of, it's been a hard market for biotech companies raising money themselves, and especially raising money behind research.
And where can people go to learn more about OpenBench and the collaborations you've been a part of?
Yeah, you can learn more about OpenBench by reaching out to me directly if you so desire on LinkedIn. You can mention that you listen to the podcast, and the Data and Biotech podcast, and heard what we're working on. I think that, if you want to check out our website, it's openbench.com, or you can email discovery@openbench.com to get in touch with me as well.
For sure. Well, James, it's been great to have you on the podcast. I really appreciate the time, and look forward to connecting down the line.
Awesome. Well, thank you, Ross.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.